Sudhakar. Today I want to say about the topical 10th class biology hydrilla experiment. What is hydrilla experiment? How this experiment will be conducted? First, aim of this experiment to prove that oxygen is liberated during photosynthesis. Actually, in photosynthesis, oxygen is liberated. So, how that oxygen is liberated? We have to test in this experiment. So, the aim of this experiment is to prove that oxygen is liberated during photosynthesis. And next, apparatus. What are the apparatus used to conduct this experiment? Means one beaker, this is the beaker, along with water, and a test tube, and a funnel, and also the hydrilla plants, small hydrilla plants, and also the glowing splinter. So these are all the essential parts which are used in this experiment. And what's the procedure of this experiment? Means actually first we have to take one beaker, and in that beaker we have to pour more quantity of water. Then we have to take one funnel. Through the stem of this funnel, we have to fix some hydrilla plants, and then this total funnel along with hydrilla plants should be dipped in the water of a beaker, and then we have to take one test tube. That test tube also completely filled with water, and we have to keep that test tube very carefully on the stem of the funnel like this, as shown in figure. Then we have to keep total this apparatus in sunlight nearly 2 to 3 hours of time. For what we have to keep outside, or we have to keep in the outside means to take sunlight. So to do that photosynthetic experiment, we have to keep outside, not in the closed room. So we have to bring, or we have to take this total experiment in the sunlight. We have to keep all these apparatus in the sunlight nearly 2 to 3 hours of time for performing of photosynthesis. So after some time, these plants which are present in the stem of the funnel, what are the plants? Hydrilla plants. So that hydrilla plants will receive that sun rays. Will receive sun rays. That means it receives like this. Sun rays will fall on the hydrilla plants. So by receiving that sun rays, that hydrilla plants will perform photosynthesis. So if we give 2 to 3 hours of time in the outside, that means in the sunlight, actually that sun rays will enter into the funnel, and then this hydrilla plants will receive that sun rays, and that means that temperature, it receives temperature. Then it will perform photosynthesis. So after performing photosynthesis by the hydrilla plants which are in the funnel, oxygen is released. Where that oxygen will go? That oxygen will move directly in the form of air bubbles into the top portion of the test tube. Top portion of the test tube. Immediately, when that air bubbles, that means when that oxygen bubbles, when they enters or they reaches the top of the test tube, automatically the water level in the test tube will decreases, and then that water will move to the beaker. Will move to the beaker. So automatically that water which is already present in beaker, that water level will increases compulsory. So here decreases means here increases. This because this water enters into the beaker. So the beaker water level increases automatically. So the oxygen will be in the top portion of the test tube. So after completion of this experiment, we have to keep our finger on the top portion or the mouth region of this test tube, and we have to take very carefully to the outside of the beaker. Then we have to keep one burning splinter on the mouth or the mouth part of the test tube. Immediately that burning splinter or that incense stick will burns very brightly, which indicates the presence of oxygen. That indicates the presence of oxygen, because in oxygen that burning splinter will brightly, it will burns. So that is the indication of oxygen. So this is the total process of photosynthesis. Actually in this photosynthesis, oxygen is liberated, that reaches the top portion of the, that means this portion of the test tube. So in this manner we have to complete this experiment. So that is the procedure. And test we know already. So by keeping our thumb here, that means at the mouth region of the test tube, we have to take out that test tube very carefully. And if we give our burning splinter at the mouth part of the test tube, that splinter will burns very brightly. That is the test tube. And what's the result? Result means oxygen liberated in the experiment. So that is the result. So to prove that oxygen is liberated during photosynthesis. So after completion of photosynthesis, oxygen is liberated. So that is the result. And precautions. What precautions we have to take in this experiment means care should be taken while removing the test tube from the stem of the funnel. So this is the stem of the funnel. So we have to take very carefully that test tube outside of the beaker. So that care is very important. So in this manner, this hydrolysis experiment we have to do for the liberation of oxygen during photosynthesis. So this experiment will ask in public examinations. So what is hydrolysis experiment? Discuss. That's the question form. Or prove that oxygen is liberated during photosynthesis. That's also one type of question. So sometimes in the public examinations, they will give this diagram, and they will ask what is the aim of this experiment. What's the aim? To prove that oxygen is liberated during photosynthesis. Next, what are the apparatus used in this experiment? They will ask like that. So beaker with water, test tube, and a funnel, and also the burning splinter and hydrolysis plant. So these are all the apparatus we are used in this experiment. And what observation we have to observe in the experiment means gas bubbles will reach the top of the test tube. Will reach the top of the test tube. So that is the observation. Gas bubbles means what? Gas bubbles? That gas bubbles are nothing but oxygen bubbles. And what test should be done in the experiment? So burning splinter test will be done in this experiment. What is the result? Means O2 is released. O2 means it's nothing but oxygen. And what precautions we have to take in this experiment? Means care should be taken while removing the test tube from the stem of the funnel. So these are all the very essential questions which regards to this experiment. So if you like this class, you please subscribe my channel. Thank you to all.